Hello student, in this video we will come to next part of class 12 biology chapter 2 sexual reproduction in flowering plant. Okay, and in this class we are going to learn or study about the structure of ovule, about megasporogenesis, and about the development of embryo sac. But before we come to this topic, let us remind something about the structure of the female reproductive part, that is carpel or pistil. Okay, and we know that this carpel or pistil represent the female reproductive part of a flower, and this carpel is composed of three parts: stigma, style, and ovary. And we also know that ovary bear one to many ovules, and these ovules remain attached to the parenchymatous cushion, which is known as placenta. And in some flower there may be a single carpel, and that condition is called monocarpellary. And in some flower there may be more than one carpel, and that condition is called multicarpellary. And in multicarpellary condition, what can happen? The carpels can be either free or fused. Here the fused condition is called syncarpus, and free condition is called apocarpus. So this is something about carpel or pistil. Now come to the structure of ovule. So there are different types of ovules. So here we will discuss a type of ovule known as anatropous ovule. Okay, and one more point, inside this ovule megaspore will be produced. Because of that, ovule is also known as megasporogenesis. So you can call ovule as megasporogenesis. Okay, now coming to the structure of anatropous ovule, we know that the main body, the main part of the ovule will be represented by a oval shaped structure known as nucellus, and this nucellus will be surrounded by one or two envelope, and this envelope is known as integument, and this integument will leave a narrow pore at one end of the ovule, and this particular region where integument is absent is known as micropyle. Okay, and opposite to micropyle you will find the region of chalaza, from where the integument arises, and this body of ovule will be attached to the ovary wall with a stalk, and that stalk is known as funicle, and this funicle will be joined with the body of the ovule in a region known as hilum. That means hilum, it represents the junction between ovule and the funicle, and towards the micropylar end you will find embryo sac, where female gamete egg is present. Okay, so this is about the structure of anatropous ovule. So in this lesson here we are going to study how this embryo sac develop inside the ovule, because in young ovule this embryo sac is absent. Okay, and in this embryo sac what is present? Female gamete egg is present. So here we will study how embryo sac develops. So to understand how embryo sacs develop, first let's see the process of megasporogenesis, because during megasporogenesis, megaspore will be produced, and this megaspore is responsible for the development of embryo sac. Megasporogenesis. So in the young ovule, what happened? Towards the micropylar end and in the nucellus, a distinct archisporal cell will be formed, which will divide to form two type of cell. One is called parietal and one is called sporogenous cell. And here the parietal cell will divide further and it become part of the nucellus. But on the other side, the sporogenous cell will not divide further. This sporogenous cell will function directly as megaspore mother cell, which undergoes meiosis to form 4 haploid megaspore. So this process of formation of megaspore from megaspore mother cell through meiosis is known as megasporogenesis. I will repeat and look at the figure. Here what happened? Sporogenous cell will function directly as megaspore mother cell. It does not divide further. Okay, and this megaspore mother cell will undergo meiosis to form megaspore. And as meiosis is reduction division, all the megaspore will have haploid number of chromosome. And as after meiosis 4 daughter cells are produced, definitely you will get 4 haploid megaspore. So this process of formation of megaspore from megaspore mother cell through meiosis is known as megasporogenesis. So this is about formation of megaspore, and this megasporogenesis will take place in ovule. And these 4 megaspore are arranged in linear form. That means they are arranged in straight line. And of these 4 megaspore, 3 megaspore which are present towards the micropylar end will degenerate and finally disappear. and thus only one megaspore will remain functional. And this functional megaspore will later develop into embryo sac or female gametophyte, okay, where female gamete egg is present. So this embryo sac will develop from a single megaspore. So the process of development of embryo sac from a single megaspore is known as monosporic. Okay. Next, let's see how the functional megaspore develop into embryo sac. Okay. So to develop embryo sac from a functional megaspore, the functional megaspore will undergo three mitotic division. And here, this three mitotic division will not be immediately followed by the division of cytoplasm. That is, every nuclear division is not followed by the division of cytoplasm. So, this three mitotic division will lead to the formation of two nucleated embryo sac, then four nucleated embryo sac, and then eight nucleated embryo sac. Okay. Then, after this, one nucleus from each side will move to the center to form two polar nuclei. And these two polar nuclei will represent the nucleus of the central cell. Okay. After this, again, the remaining six nuclei will develop into cell at the two end. And here, the three nuclei which are present towards the salazar end will develop into three antipodal cell, and the three nuclei which are present towards the micropylar end will develop into what? Egg apparatus. And this egg apparatus will consist of one egg and two synergy. That is, the eight nucleus of the embryo sac will get organized into how many groups? Into three groups. Group number one, three antipodal cells which are present towards the salazar end. Group number two, three egg apparatus which consists of one egg and two synergy. And group number three, two polar nuclei which represent the nucleus of the central cell. That means in a mature embryo sac there will be 8 nucleus and 7 cells. So how only 7 cells? Here this 7 cell will be represented by 3 antipodal cell, 3 egg apparatus, and 1 central cell. Okay. So thus at maturity, a typical embryo sac will be 8 nucleated and 7 cells. Above this, at the base of the synergy, there are some projection known as filiform apparatus. And the function of this projection is to guide the pollen tube, into the synergy. Okay, this is the function of filiform apparatus. Okay, I will repeat this point again. So, how embryo sacs develop? To develop embryo sac, what happened here? The functional megaspore undergoes three mitotic division. Here, nuclear division will not be immediately followed by the division of cytoplasm. Okay, so at last it will lead to the formation of 8 nucleated embryo sac, and these 8 nucleus will be organized into how many groups? Into 3 groups. Group number 1, 3 antibody cell. Group number 2, 3 egg apparatus, and group number 3, 2 polar nuclei. Okay, so this is about the development of mature embryo sac, and in this embryo sac, what is present? Female gamete egg is present. Okay, so this is how embryo sac or female gametophytes develop. And before we wind up, let me remind you of what we have learned in this class. So today we have learned about the structure of ovule, about megasporogenesis, and about the development of embryo sac. Okay, and that brings me to the end of today's class, and thank you very much for your attention.